Namaskaral. Myself, Manipal from Andhra Pradesh. So, regarding my experience and all, I have a total of 13 years of teaching experience in various junior colleges, and I pursued my PhD right now from Department of Botany, SV University, and I finished my PG from the same SV University Department of Botany, and I completed my PGDMB from Central University of Hyderabad, and BD from the same SV University Department of Botany. So, apart of today's class, I would like to take a small topic regarding the second year topics. Like in the second year topics, the second lesson, a very important lesson from botany, that is sexual reproduction in flowering plants. So this is a very important lesson, and in the point of need, this is a very much weightage lesson too. Around approximately we got nearly 4 to 5 questions from this lesson every year from the NEET. But as per the 2022, we have only 2 questions from this one, and 2018 also we got 3 questions. But in case of 2018 and 2017 and 2002, so where approximately they are asking about 6 to 5 questions from this lesson. So on an average, we can get 2 to 3 questions from this lesson every year. So around the weightage of this lesson totally 12 marks. Here from this lesson, the questions are continuously asking every year, but I would like to explain, as it is a short video, I am trying to explain a small topic, and which is very important, and which is crucial in flowering plants too, sexual reproduction in flowering plants too. And the students, they are a bit confusing at this topic, so that's why I am choosing this topic. And the topic which I am choosing for today's class is from this lesson, that is double fertilization. So what is double fertilization? Double fertilization is the characteristic feature of angiosperms. This is the characteristic feature of angiosperms. Exclusively, this is a feature that we can see only in angiosperms, where in gymnosperms and the lower organisms like pteridophytes and other organisms, that plants, they doesn't perform this kind of double fertilization in their sexual life cycle. Then here, what is double fertilization and why the name double fertilization is given to the phenomena? So let me explain what is double fertilization and where this double fertilization takes place in the life cycle of an angiospermic plant. See, after the pollination, the pollen grains that they are fall onto the stigma after the pollen germination, where most of the pollen grains that are being in a two-cell condition, or sometimes it may also be processed in a three-cell condition too. So most preferably we can say the pollen grains are being present in two cell condition. So I would like to explain in that way. So the pollen grains, once they fall onto the stigma, then they start germination on the stigma. So after the pollen germination from the stigma, the pollen tube is continuously developed and finally it reaches the ovary. The ovary consists of the number of ovules so these ovules are fertilized by the several pollen tubes that they are travel through the style. So here when the pollen tube reaches the ovary, that pollen tube enters into the what do you call ovule. So how this phenomenon, I'd like to explain little bit detailed here. As this is the ovule where we have the embryo sac inside, I would like to draw the typical angiospermic embryo sac here. That is a polygonum type of embryo sac. Of course, different plants they may develop different types of embryo sacs, but most of the angiosperms they are preferring they mostly possess the embryo sac like a polygonum type of embryo sac where we found 7 cells and 8 nucleated condition containing embryo sac. So here the 7 cells and 8 nucleus containing embryo sac, this is embryo sac. So when the pollen tube that enters into the ovule, it seems to be something like this. So the entry of the pollen tube, it is purely chemotoxic movement. So based on the presence of what do you call the filiform apparatus of the embryo sac, that will move towards the embryo sac. So here the same situation I would like to explain a little bit more details here. Let me draw in this way. Imagine this is an embryo sac where we have a egg and the synergids at their micropolar end region, and we have a 2 polar nuclei that are located here along with the vacuole here. When the pollen tube that will enter into the embryo sac, exclusively here the pollen tube will not be entered, that totally at this region. Of course, that will be, yes, here itself the pollen tube started degradation, something like this, where the pollen tube, which it consists of one vegetative cell and two male gametes. So this is a vegetative cell nucleus, and these are the two male gametes which are in haploid condition. Of course, the vegetative cell nucleus is also in haploid condition only. So the pollen tube dehisces at the synergids. These are the synergids. The pollen tube dehisces at the synergid area, where it releases the vegetative cell nucleus and along with that two male gametes into the synergy. So here is a very important question: where did the vegetative cell nucleus and the pollen, the male gametes are released into the inside the embryo sac? This is a question from 2013 or 14, I think so. Here from the NEET. Here in this case, we found that the releasing of male gametes into the synergids, that is the example, very important thing, where the pollen tube releases the male gametes and vegetative cell nucleus inside the embryo sac. Exactly the place they are asking. That is the question. So in 2013 or 14, I think so, that question will that question asked in NEET examinations. So here the pollen tube releases two male gametes and one vegetative cell nucleus into the synergids. Either synergids, it may be a right side synergid or it may be a left side synergid. So they are released into the synergids. And remember, at this point, now these two male gametes are gone fused with the one male gamete gone fused with the egg cell. This is the egg cell, and these two are the male gametes. One male gamete fused with the egg cell. This is what we call syngamy. We call this one as a generative fertilization. And another male gamete here, the second male gamete, which is gone fused with the polar nuclei. This phenomenon we call it as triple fusion. Triple fusion. This is also called as a vegetative fertilization. This is called generative fertilization, or what do you call germinal fertilization, we can say this one. And this is what we call vegetative fertilization. Here one more thing that is happen to see, the two male gametes are participated in the egg fertilization of egg, as well as the fusion with the two polar nuclei of an embryo sac. The fertilization phenomena occurs in two times. One in the form of a triple fusion, another one in the form of a syngamy. So double fertilization means the fusion of two male gametes, one with the egg and another one with the polar nuclei. We can say double fertilization is equals to syngamy plus triple fusion. The syngamy resulting in the formation of a diploid zygote. It is resulting in the formation of diploid zygote. Why the diploid condition is here? Because egg, which is a haploid condition, and the male gamete, which is in a haploid condition. So in syngamy, the fusion of a egg and one male gamete. So which they both are haploid in nature. So the fusion of a egg with a male gamete resulting in the formation of the zygote, which is diploid in condition. Then coming to the triple fission, here the triple fission means here three nuclei are going to be fused each other. Like one is male gamete and it is fused with the two polar nuclei which is being present in the central cell region, where until the triple fission occurs the two polar nuclei are not united. And remember this, two polar nuclei are being separately available in the central cell, right? But at the time of triple fission only, along with the male gamete, the two polar nuclei are fused together. They formed the triple fission, resulting in the formation of a pen, primary endosperm nucleus. What is pen? Pen is nothing but primary endosperm nucleus. So the primary endosperm nucleus, which is in 3N condition. This is a triploid in condition. So the triple fission is the fusion of two polar nuclei plus one male gamete. So the two polar nuclei are N plus N and the male gamete is N. So resulting in the formation of a pen, that is a primary endosperm nucleus. The primary endosperm nucleus resulting in the formation of what? It is producing, after the several divisions, it produces endosperm. Endosperm. What is endosperm? Endosperm is a nourishing tissue for the growing embryo in their early stages. Of course, based on the presence of this nourishing tissue, the seeds are further classified into albuminous seeds and exalbuminous seeds, are also called as endospermic seeds or non-endospermic seeds. Okay, so that's another thing that's I'm not concentrating on that area now. So here syngamy and triple fission together is happened in angiosperms exclusively. So double fertilization is a phenomenon where occurs only exclusively in angiosperms only. So the double fertilization resulting in the formation of a zygote as well as a endosperm, right? So the triple fission resulting in the formation of endosperm, which is triploid in condition, 3N condition, and the zygote, which is formed by the syngamy and which is a diploid in condition. So the diploid zygote is formed because of the fusion of one male gamete and one egg cell, which is both are haploid in nature, and triple fission occurs by the fusion of one male gamete with the two polar nuclei in the central cell region of embryo sac. Then what is this importance of double fertilization? You know, this double fertilization is first observed by Navashin, the scientist. Navashin, the scientist, in fritillaria, in the plant fritillaria. He discovered first in fritillaria the double fertilization. by Nawashim. Right? So this double fertilization is a characteristic feature of angiosperms. So the double fertilization, the another important thing here we can understand, the question is raised exclusively during the formation of a single seed in angiosperm. How many haploid nuclei are involved in the formation of angiospermic seed? This type of questions that might be asked exclusively based on the ploidy of the cells that are involved in double fertilization. So how many haploid cells that are involved in formation of a single seed in angiosperms? So then you can count 1, 2, 3, 4 and 5. So a total of 5 haploid nuclei that are involved in the formation of a single seed in angiosperms, right? And there is another question like how many types of how many cells that are involved in the double fertilization? Total how many cells that are involved in the double fertilization? Look at that, the total number of cells that are involved in the double fertilization is 1, 2, 3 and 4. So 5 haploid nuclei that are involved in the double fertilization in angiosperms and 4 cells. What are those 4 cells, Nana? The 4 cells are one male gamete is another male gamete, that is 2 male gametes, and one egg cell that is third cell, and one central cell, here the central cell. So totally 4 cells that are involved in a double fertilization of angiosperms. So such a kind of questions that they are asking continuously, right? So in case of double fertilization in 2018 and 2021, no, it's I think 2021 there is no question, and it is 2018 and 2017 continuously they are asking they ask 2 questions on double fertilization only exclusively, but there are no double fertilization questions asking and that particular area in this area from last 3 years onwards. But this year in 2023 I am expecting definitely a question from double fertilization area. Okay, that's why I am selecting this topic for this class. Okay, so that's the end of the class. Thank you very much.